everyone, welcome to the channel Knowledge Star, way to continuous lifelong learning. My name is Nisha Masroor. In this video, we will get to know about what is a polymer, what is a biopolymer, difference between polymer and biopolymer, and also the physical attributes of it by our expert Professor Bohidar, who is associated with National Center Excellence in Agro Nanotechnology, Terry Nanotechnology Center, India. He has 31 years of academic experience in teaching and research. His area of expertise includes nanoscience and nanotechnology, nanobiointerface science, soft matter science, polymer physics, and biophysics. Will explain to you in detail about this topic. So to get complete information, watch full video without skip.
A polymer, as you know, is basically repeating unit of a smaller entity which are called the monomers. In the language of polymer science, these are all monomer repeating units. For biopolymers, in case of proteins, these are called the residues or amino acids. For nucleic acids, RNA, DNA type, these are all nucleotides. But all of these are basically monomers of the respective polymer or biopolymer. Now, it can be a long chain unit with several or thousands or millions of these repeating units attached to each other through covalent bonding, or it can have also branches. But fortunately, biopolymers are all linear chains. They are not branched chains. Branches are observed only in synthetic polymers. So for biopolymer discussion, we don't have to worry about the branching. Now a term is introduced here, which is called the degree of polymerization, which basically counts how many monomer units are there. Okay, so that defines the polymer that one is dealing with. If you are dealing with the biopolymers, I need to specify how many residues or amino acids are there, or if you are nucleic acid, how many nucleotides are there, or how many base pairs are there. So these are the languages that are used in designating a particular polymer or biopolymer. When go to biopolymers, then we have three families members here. One is the polynucleic acid family, where you have DNA, RNA, or their derivatives. Then you have proteins, amino acid, polypeptides, and all that huge list of proteins can be made: gelatin, albumin, zen, insulin, and many others. Then there are polysaccharides, glucose, cellulose, pectin, chitosan. Okay, and our target is to make nanoparticles out of these. All right. Okay, so the same thing I've said again. Proteins basically amino acid sequence. There are only 20 amino acids. They appear in certain sequence in a protein molecule, which gives the protein molecule's identity. Whereas the nucleic acid, there are only 4 nucleotides. They form base pairs, and then they are associated in larger molecules of DNA, RNA type. And then carbohydrates have monosaccharides as monomer units. Okay, now all these biopolymers, proteins, polysaccharides, DNA, RNA, all of these are charged molecules. They carry a surface charge. Unlike synthetic polymers, all biopolymers carry a surface charge. There is a major difference between the two types of polymers. Now the comparison between a synthetic polymer and a biopolymer is, the synthetic polymers are, of course, as I've been saying repeatedly, they are not biodegradable, not environmentally friendly, not soluble in water, most of them. And they don't carry any, most of them don't carry any surface charge. They are neutral. That's the reason they're hydrophobic. Compared to that, biopolymers are all natural polymers. Protein, nucleic acid, carbohydrates, etc. All biodegradable, water soluble. And they are all charged molecules. They carry a surface charge. Okay, but as far as the physical understanding and the development of the polymer, of the underlying science is concerned, polymers are much more developed because they are easier to understand because of the complications that the biopolymers carry is not associated with them. So more generalized understanding of polymer science related to synthetic polymers is available, whereas for biopolymers, it's available to a limited extent. Okay. Regardless, overall understanding is still enough for us to process with this matter further and try to make nanomaterials out of it. Now, the physical attributes of a polymer is, as I've said, is the degree of polymerization, which is capital N here. You multiply with the monomer molecular weight, so it gives you the molecular weight of the polymer, which is capital M. Okay, so it applies to everything, polymers and biopolymers. In the case of proteins, you will multiply with the number of amino acids units there. With nucleic acids, multiply with the nucleotide weight. But to get an estimate of the molecular weight of the polymer, one is dealing with. Second one is this size. Because I'm representing them in the most common form, which is like a thread. In the language of polymer science, this is called a random coil. So I have random coil structure encapsulated in a circle that you can see here. I can take the size of the circle, which is the diameter, and give a number to my size of the polymer, or you can associate a radius of gyration value that you have studied in class 10, and you give a value to its radius of gyration, which is Rg. Whichever way, and they are all interrelated things, but if you want to give them numbers, these are the parameters that can be assigned numbers.
I hope you liked this video. If you want to see these type of videos, please do comment. We will definitely look after that. Thank you.
For more updates, subscribe to our channel, click the links shown on the screen to stay connected.